Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, hope you are enjoying good health. I am Hafiz Walidullah, lecturer in the Department of Pharmacognosy, Faculty of Pharmacy, Hajveri University, Lahore. Today we will study Ipomoea and from drug containing resins, and it is our lecture number 2. Before moving to the lecture, we will see the outline of today's lecture. In today's lecture, we will study biological source of Ipomoea and Myrrh, geographical distribution and collection of Ipomoea and Myrrh, then chemical composition of Ipomoea and Myrrh, and finally we will study medicinal uses of Ipomoea and Myrrh. Learning outcomes for this lecture. By end of this lecture, you will be able to describe the biological source, geographical distribution, chemical composition, and chemical structure of the main active constituents present in Ipomoea and Myrrh. And you will also be able to understand the therapeutic and pharmaceutical uses of Ipomoea and Myrrh. So first, we will study Ipomoea. Synonyms for Ipomoea are Radix ipome or Rhizeba jalapruit, Mexican scamoni root, and Ipomoea radix. Radix indicate a root. Biological source. Ipomoea consists of the dried tuberous roots. Remember that it is tuberous roots of Ipomoea oryzabensis ladinois belonging to family Convolvulaceae. Geographical source. The drug is collected in the Mexican state of Orizaba. Due to this Orizaba, it is given this name Oryzabensis. This is a morphological image of Ipomoea. Collection. How Ipomoea is collected? Ipomoea is perennial climbing twinner. First of all, you must know that Ipomoea is perennial climbing twinner. It produces a large, woody and tuberous root. The roots are dug, washed, cut into slices and dried, and further they are processed for use. Characteristics of Ipomoea. Ipomoea roots are large and fusiform with 3 to 10 cm thick and about 20 cm long. They occur as irregular pieces grayish brown in color with slight udder and slight acrid taste. So their shape is irregular, color is grayish brown, and udder is slight and acrid taste it has also. Chemical composition. Ipomoea consists of 10 to 20% resin. It means that the total amount of resin is 10 to 20%. Volatile oil and some fatty acids are also present in Ipomoea. The resin has an ether-soluble portion and ether-insoluble portion. So it means that resin, if we dissolve the resin in ether, some part of resin is soluble in ether and some part is not. Both the portion mean ether soluble and ether insoluble portion contain jalapin, a mixture of acidic glycosides. Ether soluble portion has jalapinolic acid, whereas in ether insoluble part it has hydroxy fatty acid, that is iprolic acid and convolvulinic acid. Uses. It can stimulate the intestinal secretion. It acts as a laxative in small doses and purgative in large doses, and it is also used as hydragogue cathartic. Hydragogue are those drugs which increase the production or secretion of water. The second drug is myrrh. Synonyms for myrrh. Synonyms for myrrh are gum resin myrrh, gum myrrh, Arabian or Somali myrrh. In Urdu it is known as marmaki. Biological source for myrrh. Myrrh is an oleogum resin. It means that it is composed of volatile oil, gum and a resin obtained from the stem of Commiphora molmol and also from Commiphora abyssinica or other species of Commiphora. Family Burseraceae. This is the morphological picture of myrrh, and it is a resinous material, and this is how it is collected from the plant. And this is the marketed product of marmaki or myrrh. Myrrh. Geographical distribution of myrrh. Northeast Africa and Arabia. Collection, how it is collected. The phloem contains schizogenous ducts and lysogenous cavities which are filled with yellowish white resinous liquid. Most of the secretion is obtained by spontaneous exudation from cracks and fissures which commonly form in the bark, and some is obtained from incision made by natives. Characteristics: Myrrh occurs as irregular masses or tears weighing up to 250 grams. The outer surface is powdery and reddish-brown in color. The drug breaks and is powdered readily. Fractured surface is rich brown and oily. Odor is aromatic and taste is aromatic also, but it is bitter and acrid. This is how it is produced on the stem of Commiphora mole in the form of small tears. This is the collected drug in the hand of a native. Chemical composition of myrrh. Myrrh contains resin 25 to 40%, gum 57 to 61%, and volatile oil content is from 7 to 17%. Volatile oil are cuminic aldehyde, alpha-pinene, limonene, and sesquiterpene, while resin acids are present in this drug like alpha-beta-gamma commiphoric acids. And third, gums yield on hydrolysis arabinose 4-O-methyl-glucuronic acid and aldehyde-uranic acid. Chemical tests for the identification of myrrh are: a yellow-brown emulsion is produced on trituration with water. Whenever we will triturate it with water, yellow-brown emulsion will be produced. And the second identification test is that ethyl solution of myrrh turns red on treatment with bromine vapors. And if this solution is treated with nitric acid, the color is purple. These are the identification tests for the presence of myrrh. Uses: Myrrh is used in incenses and perfumes. Like many other resins, it has local stimulant and antiseptic properties. It is chiefly employed in medicine in the form of a mouthwash or gargle for its astringent effect on mucous membranes. Alcoholic extracts are used as fixatives in the perfumery industry. Summary: what we have studied in today's lecture. In today's lecture, first we studied Ipomoea, and we studied that Ipomoea consists of the dry tuberous roots of Ipomoea or isobensis belonging to family Convolvulaceae. Then we studied the chemical composition of Ipomoea. Ipomoea has two parts. One is ether-soluble portion which contains jalapinolic acid, and other insoluble part, ether-insoluble part. It has hydroxy fatty acid, that is iprolic acid, and carbamazonic acid, and both part has jalapin. It can stimulate the intestinal secretion. Ipomoea act as a laxative in small doses and purgative in large doses, and it is also used as hydragogue cathartic. The second drug which we studied is myrrh. Myrrh is an oleogum resin obtained from the stem of Commiphora mole and Commiphora absinica or other species of Commiphora belonging to family Bercidaceae. Myrrh contains resin 25 to 40%, gum 57 to 61%, and volatile oil 7 to 17%. And myrrh is chiefly employed in medicine in the form of mouthwash or gargle for its astringent effect on mucous membrane. Alcoholic extracts are used as fixatives in the perfumery industry. Textbooks for today's lecture which I recommend are Trees and Evans, Pharmacognosy by Evans W.C., Textbooks of Pharmacognosy by Muhammad Ali, and third one is Pharmacognosy by Vero E. Tyler. You must go through these books and also study the chapters of resins and exclusively Ipomoea and myrrh from these books. Thank you for listening. Write any question pertaining to this lecture on the Google Classroom discussion board. I will reply to each question. Hope you enjoyed today's lecture. Assalamu alaikum wa rahmatullahi wa barakatuh. Jazakallahu khair.